Being the moderator for Breakout Session 2. Welcome. Our focus will be on complex product characterization and analysis. So for our first speaker for this session, we'll have Rosario Labruto. He is the Executive Director and Head of Scientific Affairs for Sandoz Pharmaceuticals, and he's going to be speaking on the industry perspective on the gaps in complex generic product characterization and future directions. Well, welcome, Rosario.
Good afternoon, everyone. Welcome to the next talk, Industry Perspectives on the Gaps in Complex GX Product Characterization and Future Direction. I would like to thank the FDA for the kind invitation to be able to present this talk in this breakout session number 2 workshop. In today's agenda, we're going to talk about complex GX and sources of complexity, advances in methodology for characterization comparability, and some considerations for sample preparation and CRO costs. Some things to consider during the robust design and control of complex injectable GX products, for example, are the reverse engineering and characterization, looking at it both from physical, chemical, and biophysical methods point of view. Moreover, maximizing the benefit of interaction with the FDA in regards to FDA guidances, controlled correspondences, and product development pre-submission meetings to de-risk development are strongly encouraged, and including the body of data generated during the reverse engineering and characterization during those discussions. What are the sources of complexity and challenges? We can look at it from a material complexity point of view, difficult to characterize APIs, for example, synthetic peptides or oligonucleotides. Also difficult to characterize excipients, which could be branch polymers or mixture polymers, and as well as formulation complexity and manufacturing process and supply chain complexity. Supply chain complexity, for example, where multiple process unit operations extend beyond one facility, if not vertically integrated, such as a drug-device combination product with manufacturing at one site and drug assembly at different sites. If we take a deeper look at analytics complexity, it's how do you demonstrate that the API that you do have, which is complex, is the same type of complex API that's used in test product RLD. This is done by characterizing the intended API, the test product, as well as the RLDs. Looking at from a variety of perspective, physical, chemical, impurity profiling, identity, primary structure, high order structure, aggregation, just to name a few. And what is key is the intense reverse engineering. Also, the health authority interactions to align on acceptable in vitro study designs. Strong scientific justification for those in vitro study designs are absolutely needed for an in-depth understanding of the API chemistry and interaction with formulation components and critical quality attributes affecting efficacy and safety. It does require demanding characterization methods and orthogonal methods for complex molecules like peptides and oligonucleotides, for example. And it requires primary as well as orthogonal techniques. If you take a look at aggregates, and this is both for covalent and potentially non-covalent type of aggregates as well. Additional sources of complexity could come from access to multiple lots of RLD and or different ages, lack of readily available in-house characterization tools or expertise, also complex, expensive, and orthogonal analytical techniques as well, and leveraging on external CRO capabilities and interpretation of the data, as well as removing or minimizing spectral interferences and the appropriateness of the sample preparation. From a regulatory complexity point of view, product-specific guidances, when available, are very good starting points. However, sometimes these guidances may not be available due to long lead times, and general guidance, even when available, for example, as the liposome guidance, may not provide sufficient clarity, and replication of experiments from published literature could also prove to be challenging. Moreover, guidances may also evolve subject to new technical information or a citizen's petition submitted by innovator companies. So let's take a deeper look at advances in methodology for characterization and comparability. The nanodisc system, dissolution for nanoparticle formulation, is a new system based on cross-flow filtration and it's combined with conventional dissolution apparatus and with the aim to automate the filtration process for nanoparticle formulations. It can address insufficient separation of nanoparticles from the dissolution medium during the sampling process, and other advantages include CFF to separate these nanoparticles from the dissolution medium and can prevent the blockage of filters in the dead-end filtration. Another interesting technique is sedimentation velocity analytical ultrasonification in the development of nanoparticle drug products used for size distributions analysis. Sedimentation coefficients proportional to the molecular weight and hydrodynamic radius, assuming the samples have a similar hydrodynamic shape, and it allows for comparative characterizations of iron colloids. For example, taking a look at the SPAN and the D50, comparing the brand versus interradiator in this particular example. In this particular example, a variety of other attributes were compared and they were similar. However, when the brand and the generic complex product were looked at by looking at the molecular weight distributions by gel permeation chromatography, they were the same. However, by this particular technique, they observed minor differences. And then these particular differences did not, though, impact the biological activities in separate studies of in vitro cellular uptake and rat biodistribution. That is why it's important to take in aggregate, no pun intended, all characterization data obtained using the various primary and orthogonal techniques. In the next technique, small angle X-ray scattering, it can be used for ferric nanoparticles. And in this particular technique, the measurement results of the DLS and SAX measurements complement each other well, since DLS can capture the whole particle, including the shell, while SAX determines the particle size as well as the particle shape. This enables the user to differentiate between the core and shell dimensions of the particle. And for this specific example, it shows that both iron preparations actually have a cylindrical shape, while iron sucrose can be described as a long cylinder with a length-to-width ratio of 4.3, and ferric carboxymaltose in this particular case can be described as a short cylinder with a length-to-width ratio of 2.4. In the next example, SAX and SANS for the characterization of liposomes are really great techniques for taking a look at, for example, a higher space and time resolution for in-depth structural characterization. SAX and SANS scatter techniques provide information at the nanoscale. These techniques with higher space and time resolution for in-depth structural characterization of liposomes provides additional characterization information, and when combined, allow a fine structural description of the particles and allow to characterize internal aqueous environments of liposomes. SAX being more sensitive to the polar layer, while SANS provides more information on the hydrophobic layer. Characterization of liposome formulations for size-based distribution of drug and excipients using AF4 and LCMS. AF4 is used on the front end, and LCMS is used on the back end. Here, basically, LCMS was taking a look at the lipid DOX composition, and the drug-lipid ratio was close to be constant as a function of particle liposomal size, suggesting drug loading was proportional to membrane surface area of any given liposome particle in this particular generic formulation being developed. And the last example, NMR, peptide higher order structure is a quality attribute that could affect therapeutic efficacy and safety, and higher order structure differences can be quantified using NMR and PCA, principal component analysis. The unitless Mahalanobis distance, DM and PCA space, can be calculated between innovator reference listed drug and the intended follow-on product. These studies support the use of NMR as a robust, reproducible, and sensitive method in identifying higher order structure changes. Generic products of the same drug substance could cause differences in the peptide HOS through subtle pH or ionic strength variations. Therefore, separating buffer components induced HOS changes from drug substance manufacturing-related HOS differences may be difficult, but need to be considered in the sample preparation and analysis. In regards to sample preparation, additional considerations, the type of dilution, how the product is diluted, whether dialysis needs to be performed or buffer exchange, and other factors need to be considered for a good comparative analysis between test product and RLD. And CRO testing costs should also be considered as part of the development plan. What is the CRO or CROs that will be approached to run the analysis? How many samples will be analyzed? How many replicates will be performed? Will the report be included? And various other types of questions should be considered in choosing the adequate CRO. And to close, advancing complex GX is a challenging task. As we may know, there is a significant number of off-patent complex product drugs for which there are no or few generic versions, and larger number of newer complex drugs where no PSGs exist. Therefore, to bring new complex generic drugs to market requires access to the appropriate physical chemical characterization methods, both primary and orthogonal, the access and availability instruments, the right experience, availability to the R&D, and also leveraging on FDA guidances when they do exist, as well as interaction with the FDA via product development pre-submission meetings, and leveraging on active FDA research and publication of published literature. From an industry perspective, there are some opportunities. In regards to product development meetings, could there be an easier process to submit and request a meeting? Questions only in the initial request, and once the meeting is granted, then a full package is delivered before X time when that meeting is to be held? In regards to product-specific guidances, could there be a reduction in lead time, issuances of product-specific guidances after X time from complex product NDA approval? And lastly, for mid-cycle review meetings, could there be a dialogue enhancement to address outstanding scientific questions during the mid-cycle review meeting? By active work in this complex area, this will allow for greater access to medicines for patients. Thank you for your kind attention and looking forward to an interactive and robust discussion during the panel discussion. Thank you.
All righty. Thank you, Rosario. That was an excellent talk, and you brought up some fascinating new techniques and technology for consideration for our approach to looking at generic products and looking at how we can analyze them. I'm going to open your talk up for discussion. First of all, I'd like to introduce our expert panel. And I'm going to, we have quite a few actually. So I'm actually going to ask folks to say who they are when they speak, and also for folks to just briefly say your affiliation. So we have Kevin Hawkins, we have Darby Kozak, Likhan Liang, Malaswala Reddy, Pahala Simamura, Ram Randad, Ravi Patel, Rosario Labruto, and Adama Rao, and Rachel Dunn as well. And so I think we're getting all our videos up right now. Folks, you can, so you can see us. Let's start our webcams up. There we go. I think you can see, uh-oh, I have the wrong webcam here. Okay. Great. And you can see I'm kind of backlit here, but okay. So with that, I'm going to address our first question up for our panel. So you heard our speaker talk about some of these new methods. Can folks identify what are some of the barriers to implementing some of this new technology, in your opinion? Common issues include not implementing the right system, failing to get a regulatory nod or the company nod to getting them moving forward, or just the fact of the newness of the technology. So any thoughts on that? Any volunteers to address how some of the barriers to implementing some of the technologies that were brought up?
Yeah, I mean, one of the barriers is really, there's some of the techniques are newer, right? So, what are the right CROs to approach? And in regards to some of these techniques as well, too, what is the right body of qualification or validation, even if it is required, and instrument calibration? I know there was a presentation by Cameron Smith, and there's also an article as well too around this. However, more clarity, I think, Markham, is needed around this, because it can be extremely challenging. Like, for example, if you have SACs, or you have AUC, how are you actually going to be, doing a calibration for it? I mean, I know with AUC, possibly you could be using some type of, gold particle standards or some other polystyrene standards. Maybe, do we really need to go that deep? Hear other people's thoughts on that. Thank you.
Dama, I saw you shaking your head. You look like you were in agreement. Any thoughts there?
Yes, Markham, I think so. What Rosario is mentioned is right. So, very important thing for all these complex products characterization is the right analytical technology and the right skill set of the scientists to look at the data. And starting from calibration to the sample preparation, particularly, for example, if you take the SBIA, where even the cell cleaning is a skilled art, and then followed by the sample loading, and then so getting the right data and the interpretation of the data is also very, very critical to understand the quality of the product per se. And these are the complex technologies which are evolving a lot. I think right skill set at the right time is important. Otherwise, we'll end up with so wrong conclusion.
So Dama, I don't know if folks understood. Dama, your microphone is clipping a little bit, but I think what Dama is saying is that the data that you're looking at when you get a new instrumentation, and it's going to be very important to make sure that that data is in the right ballpark at least. That you'll want to run checks against your prior systems, etc. I think when you get a new instrumentation and you're looking at data, you're going to want to align with your previous systems to make sure that you're not totally out of whack. And then when you're looking at the data going forward, you're going to have to figure out how relevant is that, what those additional decimal places might earn you, and for certain systems, and other systems, what is the relevance of that data in the context of what we're looking at. I want to ask Darby and Prahala about the relevance for bioequivalence for these products. When we look at new instrumentation, say we're looking at a Raman particle size or a Raman spectroscopy hooked up with particle measurements, what kinds of things are we looking at regarding adaptation of that technology for regulatory purposes, for sameness, etc.?
So this is Darby. I can actually start and kind of go with the general sort of question that was posed a little bit earlier. I think FDA is always very interested in new analytical methods and more sensitive techniques. One question I think all our reviewers are looking for, and the key aspect is it fit for purpose? And as well as what's the key question that you're trying to answer or support through that method? And so there's multiple ways that you can engage the agency, but that's kind of the key sort of thing that we're trying to better understand. And there is instances where the calibration or that bridge back to a calibrant, even though it might not necessarily be 100% sort of the correct bridge, helps us to kind of better understand how the technique works and things like that. I think Rosario mentioned the sedimentation or the AUC, the ultracentrifugation method. Like you said, as the gold particle, at least there is, we can see that in the hands or use of the technician that you have, they're getting the results that they expect. So that way, I think it's not necessarily calibrating the instrument per se for the liposome analysis, because liposomes are very different than gold particles, but it's showing that the analytical technician that's using the instrument understands the instrument and can show us the fact that they're being able to do that sort of component. That's not always 100% like the key aspect, but it does give us that sort of additional sort of bridge of understanding. and kind of that aspect to start that sort of discussion and to build that reliance upon the data that's being presented that much further. So I think that's one of the key sort of aspects, and there's multiple ways I think we can engage with the FDA through that. One of the things I think you mentioned also is through the pre-ENDA meetings or even pre-submission meetings. These are good aspects to kind of explain that new technology, both in the development aspect and then just before your submission, kind of explaining how you built that bridge and why this technique is going to be beneficial for the ultimate aims of demonstrating either pharmaceutical or bioequivalence of that drug product. I let Pahala and a few of Ram and some of the others sort of.
If I may, I just want to.
So Darby, sure. Sorry, Pahala. I just want to clarify, Darby, on that point regarding the pre-ENDA meeting, do you think the pre-ENDA meeting time is the best time to bring up new analytical technologies?
So I think there's a couple different sort of routes. The pre-ENDA definitely gives you a dialogue sort of perspective in the development meeting, because you're going to develop your analytical method, discuss how it's going to be used. And this is even for new analytical, new modeling, any type of thing that you feel could have a good conversation, both asking FDA's questions on it, as well as to get and educate FDA more on what your thinking is and rationale. So I do think that that gives you one of the better aspects to engage us.
Yeah, I was going to say,
Paula, you were going to say.
Rosario had a good point. But again, for something that is new to be used to characterize complex drug product, right? So there are a lot of complexities to it, a lot of unknowns, if you will. But I think currently there's some techniques that are commonly used. I guess my point is, if we kind of like have this develop and use as a complement to what we, what we are currently use, I think that would kind of like a nice addition to, in order to understand, to characterize that particular drug product.
Yeah. If I could just maybe add on to what Darby said, right? So the question becomes, if I go back to that article, the use of two different calibrants, right? Could time be well spent maybe looking at some other parameters? Is truly two different scientists needed? As you may recall, most companies, even large companies, may not have that instrumentation. So you're paying for instrumentation time, access to the instrument, and availability. So, just thoughts around that, value added, I think is going to be super important. Doing robustness for a characterization technique, if you can demonstrate already some type of selectivity or sensitivity, is robustness truly needed? Just food for thought. I would love to hear from others around that topic, please.
Ram? You look like you're getting ready to say something, but you're on mute. You're going to have to.
Okay. I did unmute. Rosario, very first point, what he brought up in his talk was reverse engineering. And as Darby said, FDA is very encouraging of new technology, but is it relevant? And does it answer the question? So let's just spend a minute or half on the reverse engineering aspect of it. It is very important to understand the reason of complexity. Is the complexity due to API? Or is it a complexity related to the product or the formulation? And when you look at the API, if the complexity is of the API, is it a natural product or is it a synthetic product? If it is a natural product, whether it is a mixture. And I think what I have noticed is most of the industry stops at, it is a natural product and it is a complex product. But I think we need to go one step further and try to understand the biology. What is a period for collection? What is a harvesting period? Where does the API should come from, whether it is the stem or the leaves or the flower, or if it is coming from the animal source, what is the source, what is the biology of a particular life cycle of the animal, and what should be an harvesting period. It is very important to understand the biology, and I do want to drive this point home if you are particularly working on the natural product. Once you understand the product, and many times you might find that the manufacturing process will have to be designed to mimic the RLD. It is possible that you may not get the product just from an extraction or the manufacturing process. It may require certain blending. So reverse engineering and the analytical methods tied to understanding the components of the API becomes a very key factor, along with the biology and the implications of those, whether you would call impurities or concomitant compounds. Since if it is a complex API, it is very difficult to define what is an active. So maybe everything
exactly,
is an
Ram.
active.
So you're talking about the natural substance.
Yes.
Right.
Well,
Thank you, Ram.
even in synthetic, synthetic polymers, Markham.
Sure. I wanted to move on to folks who haven't had a chance to speak or express their opinion on this panel. Ravi, did you get a chance to speak yet? I don't think so. And Malaswara?
Yeah. So Markham, I just wanted to add
Ravi,
one.
you are still on mute. Yeah.
Yeah. Markham, shall I go?
Who's speaking? Hello?
This is Malleshwar.
Go ahead and say who you are.
Yeah. So Markham, I just wanted to add.
Malaswara, go ahead.
Yeah, I just wanted to add one point here regarding the pre-development meeting and the timing of using the orthogonal techniques, whether we should upfront go and tell agency that I wanted to do this multiple orthogonal techniques. Is that useful? Whether I should be using or not. So I think when we are discussing with agency, it is important that you understand the RLD and you assess what is the key characteristics required. But also parallelly, you also try to do some research on your product and try to see whether while you are manufacturing your own product, you are able to get those characteristics using those techniques. If your research is not in a reasonably understandable stage, then you will be only theoretically presenting to FDA. It will not be very useful. So
that's an excellent.
Yeah. So what we understood is if you generate some data and you found it useful, then it will be very useful.
Yeah, that's an excellent point. Part of our research is looking at making our own products. We're looking at the novel technology and trying it out and looking at how useful is it in the context of what we do and rendering a regulatory opinion about the utility of that device or product as it's being used. Ravi, are you able to work out your sound issue? And then we'll go back to Kevin. I think, Kevin, you wanted to say something as well? Go ahead, Ravi. Ravi, you're still on mute.
Sure, I can go ahead in the interim and give Ravi a bit more time. So hi, everybody, and thank you, Rosario, and everybody for the discussion. I think I just want to kind of take it a little bit back to Markham's original question, which is what would hinder generics industry in some of these new techniques and adopting those techniques. And I think the points that have been raised have been really good, and it's a blend of all of them. You know, the techniques are often new and not necessarily understood, and certainly we don't necessarily have experts within the industry, within the company, that might fully understand that technique. And then when you blend that with the calibration or the robustness type issues that give you a lack of clarity, and you're faced with knowing that your product will always be slightly different, but trying to be able to have the confidence that you can justify the differences that will be thrown up makes it reluctant at the start for many companies, but it's essential that they take that research work, as Ram explains. You know, you have to characterize your products up front. You have to really make sure that you're matching the RLD right back to the source of the API or the source of polymers in long-acting release, or it's fundamental that you understand those and take up these techniques. But the sheer amount of uncertainty sometimes makes it difficult for companies to start, and that uncertainty is, for simple products, it's very clear. You're looking for a few impurities, you're looking for slight changes in formulation, maybe some slight changes in simple excipients. But when you start adding complex APIs into complex excipients, into complex products and complex devices, and then throw on top of that a new complex range of analytical techniques, as Rosario explains, you're not looking at two techniques, you're looking at all of them. And it's that uncertainty that I think slows down the industry.
Exactly. Thank you, Kevin. I see Ravi has unmuted his mic successfully, talking about use of technology. Ravi, go ahead and chime in. We do want to hear your opinion.
Yeah, thank you. Mark, I just wanted to add on to those Raman technologies and all those things, right? Where specifically we were talking about the particle size measurements, and Rosario brought that point that we really have to go about this selectivity, sensitivities, and all this. Yes, we're talking about the complex products where we have multiple APIs, mixtures in the product. At times, I think yes, use of Raman technologies where Dama also got the good point that using right technology at right times where an article.
Ravi, I'm going to interrupt you, Ravi. Yeah, as you can see, our transcriptionist is having trouble hearing you as well. It sounds like there's a lot of noise in your microphone.
Okay, I'm sorry. Then I'll skip on.
One option is for you to type in your input on the, and we can read it. All right, Darby, go ahead. You have a comment.
I kind of want to go back to Rosario's additional sort of comment about the robustness and understanding and all the differences that need to be done there. I think one of the uniquenesses about today's workshop in particular is that you guys can propose what types of research that you want to do for funds to be spent on. And so if there's a promising new analytical method, it's always great to be able to say that, hey, here's something that we think that would be beneficial for the FDA to better understand, get a better sort of grasp of, look at some particular products, understanding the limitations and its potential use, and that then brings some of that in-house information to us. The other aspect, like I said before, is if you as industry want to develop that, engage us through those PDEV meetings or product development meetings and things like that to help educate us in that sort of aspect. But I think that's one of the unique things about today is really once we have a better grasp of the technique, its limitations, its potential fit for use, we can then provide the review and the information back and forth and have those really substantive scientific discussions when assessing the application and providing hopefully that guidance, that feedback. So I think that there is what I wanted to kind of a good take home.
All right. Well, thank you very much. This ends this panel, and then we're going to go on a short break, is my understanding. Is that right, Mark? And then we're going to hear Dr. Kozak speak on behalf of the FDA, aside for this panel, and then we'll go into and then batting cleanup for the talks are Malaswara, and then we'll back to a whole panel. After each talk, there'll be panels, and then we're going to have an end panel where everybody gets to chat a little bit about what we've all heard, and we can bring in discussion from this morning as well. Okay, Mark, take it away. We're going to go on break for a short while, 10 minutes.